Hello everyone and welcome to Instant Biology by Dr. Neelam. Again, I am here with you with a very important topic, that is biosensors. So for those of you who understand Hindi better, I have already made a video on this particular topic in Hindi. You would get the link in my playlist and I'll also paste the link in the description. So you can study this topic in Hindi in that particular video. For those of you who understand English better, I would be talking about biosensors in this particular video in English language. Okay, so without wasting much time, let us move on to what are biosensors and details about biosensors in this particular video. So biosensors, firstly let us understand what biosensors are in very easy language. Biosensors are nothing but they are measuring devices. Okay, so let us start with this. Biosensors are nothing but these are measuring devices. What do you mean by measuring? In case of or through a measuring device, what would you do? You would measure something. What would you measure? Now, to answer that, what would you measure? We are coming to the next portions. So, measuring device will help you to do the measurement. Now, what do we measure? It converts biological responses to electrical signals. So biosensors, what is the function of biosensors? It will convert biological response to electrical signals. Now what exactly does a biological response mean? Let us understand this with the help of an example. Suppose this is a, let us say this is a enzyme. Okay, let us say that this is a enzyme. I am writing E for the enzyme. Now suppose this enzyme has an affinity towards a substrate. Let us say that this is a, or let me just rub this and change the shape. This suppose is the substrate. I am writing S for substrate. Now, if this enzyme and substrate will form a complex or they will interact with each other, then definitely what will this be called as? This will be called as a biological action. Or you can also say that it will be called as a biological response, because enzyme and substrate will be interacting with each other, and what they will form is they will be forming a product. Okay, so what does this mean? So this clearly means that we can measure with the help of a biosensor, we can measure a biological response by converting it into electrical signals. Now, when you convert a biological response into electrical signals, that means when an enzyme and substrate they will interact, a product will be formed. This will be communicated to us in the form of electrical signals, and electrical signals are easily displayed on a particular monitor by signal analysis unit, right? So these electrical signals we would be easily be able to interpret on a monitor or a display unit through the signal analysis unit that is attached. So whether a substrate or an enzyme is interacting or not, or let us say whether an antibody or an antigen is interacting or not, and many such responses, you can easily analyze, you can easily understand whether the response is being created or not by converting these biological responses into electrical signals and then reading or analyzing those electrical signals using our monitors or using other mechanisms. We would be understanding this in much more detail in the upcoming slides. Now, what is the definition of a biosensor? Biosensor is self-contained. What do you mean by self-contained? Self-contained simply means that it is a unit basically. That means it has all the required things. Biosensors has multiple units attached to it, but it is a complete setup that will be able to, that is a self-contained, you do not need anything else. This is a self-contained or a self-sustained unit that will be working in order to give you a desired result, give you the result whether a biological response is taking place or not. So what does self-contained means? It is just one big unit, and this will be telling you analytical device, self-contained analytical device. Why are you calling it as an analytical device? Because with this device you can do analysis. Okay, for example, over here we can analyze whether the substrate is interacting with the enzyme or not, or how much substrate is present, how much reaction is happening. All of the analysis can be done. Now, analytical device that incorporates a biologically active material. So in the biosensor, there should be the presence of biologically active material. Why should there be presence of biologically active material? Because this biologically active material will only be responsible for the biological action or biological response. So this biologically active material should be present in contact with the appropriate transducer. Now this transducer is a term that is related to electronics. Basically, transducer, what will it do? It is responsible for the conversion of biological responses into electrical signals. This is exactly what the transducer does. Transducing means converting. Converting what? Biological signal into an electrical signal. So this will be done by the transducer for the purpose of detecting the concentration or activity of chemical species in any type of sample. So when the transducer converts this, what is the purpose that it fulfills? Now with this electrical signal, you can detect the concentration as well as activity. I already told you, you can analyze whether the substrate is present, and if the substrate is present, what amount of the substrate is present, and what is the activity of the enzyme. This everything can be understood by using a biosensor. So I believe this would be a clear-cut concept. Let us understand this once again. So a biosensor will be having a biological biologically active material, and it will also be having a transducer. The transducer will be responsible for converting the biological result into electrical signals, and these electrical signals will be giving us the information what is the quantity of a particular it will help us detecting the quantity of a particular thing, or the response or the reactivity or the activity or the function of a particular thing. So this is the concept about biosensors. Now let us move on to the next thing. Let us take an example, or let us take the help of this particular figure and understand how a biosensor works. So this is a sample. You can consider this as any sample. Now this sample can interact with, now let us say that over here an antibody, or let us say an enzyme is present, or something is present. So let us say these are the enzymes that are present. And what is this? This is sample. Let us say that this sample is comprising of the substrate. Okay. So now what I am trying to say is that we are trying to show a biological reaction that might happen. Suppose we are having biologically active material. This was the part of biosensor. What are the examples of biologically active materials? You can either use enzyme, you can either use microenzyme, you can either use immunoagent, chemoreceptors, or tissues. You can use any of them. They will be considered as bioreceptors, and they are the part of biosensors only, right? Now when these things, or when these bioreceptors will come in contact with the sample can be anything. For example, if we are taking an enzyme over here, we can consider the sample to be as a substrate. If we are creating a microorganism over here, then also we can consider this as a substrate. Or if you are considering an antibody, you can consider it as an antigen. Anything you can consider, but the thing is that both of them should be reacting with each other, right? So this is the bioreceptor, this is the sample, and suppose we want to understand that whether both of them are reacting or not using a biosensor. So this is the part of biosensor. Now if the sample and the bioreceptor, they are interacting with each other, what will happen? A biological signal will be transferred. What will happen? A biological signal, let me write over here, biological signal will be produced. A biological signal will be produced, and this is the biological signal that is being produced, and it will be perceived by transducer. Now there can be different types of transducers. So what are these? These are transducers. Transducers can be electrochemical based, optical based, piezoelectric based, calorimetric based. So these are the transducers, and what do the transducers do? They convert the biological signal into electrical signal. So biological signal will be converted into the electrical signal over here. Now this particular signal, or the electrical signal we should say, is now transferred to the signal processing unit. What is the purpose of this unit? It will be helping to analyze, it will be helping to display the electrical signal in a more readable format, in a more readable way, so that we can understand it better. So in this way, we would be able to see the reaction, what exactly happened in the biological sample, whether it was reacting with the sample or not, and what was the reaction we can finally read over here. So this is basically a flow diagram of how a biosensor basically works. Okay, so I believe this would be clear. Now let us move on to the next thing. This is another diagram that is basically depicting the same thing in a much more understandable way. So let us start this, understanding this. You can see over here that we are starting with analytes. Analytes means the things that you want to analyze. It can be anything like chemicals, pollutants. If suppose, let us say, chemicals or pollutants are present in water, or let us say in the air, how would you detect it? You can use, or you can detect it using some of the biosensors. Suppose in the biosensor, the biologically active part or the biologically active material has enzymes or DNA or RNA or antibody or antigen having anything, and these have a strong affinity towards the chemicals or pollutants. So what will happen? A biological reaction will be taking place. Biological reaction will be taking place. Biological reaction will be taking place between the sample and biologically reactive material. Biologically reactive material. So what is the biological reactive material? This is the biologically reactive material. This is the sample. They both will interact, and then what will happen? There will be a signal that will be produced. That would be a biological signal. But now this biological signal has to be converted into the electrical signal. Who will convert it? This is the purpose of the transducers over here. So transducers can be fiber optic based. Transducers can be electrode based. They can be any type. Now, utilizing these transducers, the electrical energy or electrical signal will be produced, and finally the electrical signal will be amplified, and signal processing unit will be there. Signal processing unit will send us the electrical signals. In this way, we would be able to understand whether the biological reaction was taking place or not, and all the other things can be analyzed and accessed and can be understood in a much better and useful way. So this would be clear. Now let us move on to the next portion. What are the different types of biosensors? I have already explained it using a diagram. Now let us understand the theory that is connected with the biosensors. So, parts of biosensors. Biological detection system. So what is the first part? We are discussing the parts of biosensors. The first one is biological detection system. I have already explained it to you. I said that there has to be a biologically active material that should be present inside the biosensor, whose function would be to interact with the sample. So this is what is written over here. So bioreceptor can be anything like enzyme, antibodies, small binding molecules, living cells, organelles, anything that is related to this. Okay, so these would be called as biological detection system. Next part is transducer. I have already explained it to you. What is the purpose of transducer? It converts interaction, that is the signal, biological signal, into measurable signal, that means the electrical signal. So conversion of biological signal into electrical signal, this will be the function of transducer. Next, signal processing unit. What is the purpose of signal processing unit? Convert signal to workable form, more understandable form we mean. Okay, so these were the basically parts of the biosensor. Let us move on to the next thing. Next, what are the types of biosensors? Now there can be different types of biosensors, primarily two classes of biorecognition process. That means you can classify the biosensors into two classes on the basis of the biorecognition process. What is the way of recognizing a particular sample? In this, considering this biorecognition process, you can divide your biosensors into two parts. What are those two parts? The first is bioaffinity recognition. The second one is biocatalytic recognition. Firstly, let us understand what is bioaffinity recognition. In case of bioaffinity recognition, the interaction between the analyte and receptor, is very, very strong. What do you mean by analyte and what do you mean by receptor? Receptor was present on our biosensor. That means the biologically active material was the receptor. and the sample was the analyte, because we have to analyze whether it is present or not, and something like that. So analyte and receptor binding is very strong, very strong. Next, transducer detects bound receptor analyte pair. So the function of transducer will be to detect this strong binding of analyte and receptor. So this is called as bioaffinity recognition, because the analyte and the receptors have very strong affinity towards each other. This will be the function of transducer, to detect this affinity. Next one, biocatalytic recognition. What do you mean by catalysis? Catalysis means a reaction has taken place, right? So analyte is chemically altered to form a product. Now what happens over here? Over here, no strong bonding is seen between the analyte and the receptor. There is no strong bonding. But what is happening over here? The receptor, it is when the ligand or when the analyte is binding to the receptor, what is happening? The receptor basically modifies the structure of the analyte, or it is chemically altered, it is chemically changed. The structure of analyte is chemically changed, and it is led to the formation of a product. A new product is formed, and this particular product is being detected by the transducer. What is the function of transducer over here? It will detect the product that is formed over here. So I believe this concept would be clear. So the second part is biocatalytic recognition. Let us move on to the next part. The next is types of transducers. We have to now discuss about the different types of transducers. Okay, we have discussed about the classification of the biosensors. Now what are the different types of transducers? Transducers can be categorized into multiple types. First of all, electrochemical transducer. What do you mean by electrochemical? Electrochemical transducers can be further given as examples of amperometric and potentiometric transducers. What do you mean by amperometric and potentiometric transducers? Amperometric, basically over here you should understand that it is something related to potential. What is happening over here in case of amperometric transducers? What is happening? Two electrodes are present and a voltage between these two electrodes is fixed. This is what is written over here. A potential or voltage between the two electrodes is set, or it is fixed. Now what happens is the current is produced by oxidation and reduction of an analyte. What happens is when the analyte comes and binds to the receptor, what happens is the analyte is oxidized or reduced. Now what do you mean by oxidation or reduction? In case of oxidation or reduction, the H plus ions are given off, or it is accepted. What will this lead to? This will lead to the flow of electrons. Okay, this will lead to the flow of electrons, and since the flow of electrons would be there, current will be produced. So what is happening, or why is the current being produced? Because of the oxidation or reduction of the analyte. Who will do the oxidation or reduction? This will be done by the receptor. Next, this change in the current, or the current that will be produced, would be measured. How can it be measured? Because since the potential between the two electrodes is already fixed, any kind of current that will be produced will be altering this potential. If this potential is altered, our transducer will become activated and it will send the signal to the signal processing unit that yes, some kind of reaction has taken place, right? So this is correlated with the concentration. That means if more current is produced, more concentration would be there, so on and so forth. Now let us move on to the next type of the transducer, that is potentiometric transducer. Of course, this clearly means measure the electric potential due to the changes in the distribution of the charges. Potentiometric also basically has the same concept. Over here it is basically measuring the electric potential. Over here what we were measuring is, we were measuring the current that was being produced. Over here you are measuring the electric potential due to the changes in the distribution of charges. Of course, because of the oxidation and reduction, charges would be changed. Since the charges are changed, we can be measuring, or we would be able to measure the electric potential, right? Now this was clear, so let us move forward on to the next slide. The third part, or the next type of the transducer, is the calorimetric or thermometric transducer. This is clear by the name itself. What do you mean by calorimetric? Heat of biochemical reactions. Now you would be knowing that many biochemical reactions are exothermic or many are endothermic. You can connect them with the Gibbs free energy. For the reactions that have Gibbs free energy as negative, or delta G value as negative, these are exothermic reactions and will give off heat energy, whereas some reactions are having delta G positive value. These reactions will be giving, or will be taking energy, taking in heat, and therefore they will be called as endothermic reactions. So the same thing is being mentioned over here. Heat of biochemical reactions will be measured by calorimetric or thermometric transducers, and this will be exactly proportional to the analyte concentration. More heat is produced, more is the analyte concentration, so on and so forth. Now, let us move on to the optical transducers. Optical transducers will definitely be linked to the light. So what? Chemiluminescence, fluorescence, light, absorbance, phosphorescence. So these are all the techniques that are related to optics, basically to light. So obviously optical transducers will be accessing or will be using any data that is related to the light, any change in light any change in light intensity or something like that. Okay, so this is optical transducers. Now let us move on to piezoelectric or mass sensitive transducer. Multiple times this type of transducer has been asked. So what do you mean by piezoelectric? The piezoelectric mass sensitive transducer, basically what is over here is that a bioreceptor is connected to quartz crystal coated with gold electrodes. Again, so a bioreceptor, let us write it like this, a bioreceptor, it is linked up with quartz crystal coated with gold electrodes. So let me write it as P, piezoelectric transducer. Whenever this bioreceptor is interacting with the sample, so what is this? This is the sample. So whenever this will be interacting with the sample, what will happen? Definitely there would be some change or there would be some load on this bioreceptor. There will be some mechanical stress that will be present in this bioreceptor. This mechanical stress will be converted into electric current by this piezoelectric transducer, and this will be sent to the signal processing unit. Okay, that means the mechanical stress or weight or load is basically converted over here. In case of piezoelectric or mass sensitive transducer, any change in the mass, any change in the mechanical stress will be converted into electrical signals and it will be carried forward. This was the concept related to piezoelectric or mass sensitive transducer. I hope this much is clear. So we have completed the different types of transducers. Now let us move on to one of the examples of a biosensor device. What are the examples? Let us understand this. Enzyme based glucose biosensors. Now you would have seen glucometers, or you might be having glucometers inside your house. Glucometers are nothing but these are the biosensors, enzyme based glucose biosensors. Now, how do you use a glucose based, or how do you use a enzyme based glucose biosensor? What is the mechanism of a glucose biosensor? So over here, glucose plus glucose oxidase. Glucose oxidase is having a FAD, basically it is having a cofactor FAD with it. You might be knowing what cofactors are. So glucose plus glucose oxidase. Glucose oxidase is an enzyme. This is a enzyme, and this enzyme has a cofactor FAD. So when glucose and glucose oxidase are reacted, what happens is glucose oxidase gets reduced. So over here you can see that FAD is getting converted into FADH2. What does this mean? FAD is getting converted into FADH2. What does this mean? This means that reduction of glucose oxidase has taken place. So glucose oxidase over here was oxidized, over here it becomes reduced. And what about glucose? Glucose has become oxidized because gluconolactone is formed. Gluconolactone is nothing but it is having a ketonic group. Gluconolactone is nothing but, you can also understand with the term, gluconolactone is having a ketonic group. What does glucose have? Glucose is having multiple OH groups. So OH group is now converted into ketonic group. Obviously what has happened is oxidation has happened. You have oxidized glucose into gluconolactone, and what you have done is you have reduced glucose oxidase into glucose oxidase FADH2. FAD has been converted into FADH2. Now, glucose oxidase FADH2, when it is reacting with oxygen, why would it react with oxygen? Because it has been reduced. It would want to give its electron to a final electron acceptor, to an electron acceptor. What is the electron acceptor over here? It is oxygen. Oxygen is present in abundance in the atmosphere. So glucose biosensor, when oxygen definitely is present in the environment, this FADH2 will be reacting with oxygen and it will be donating its electrons so that it again gets oxidized. That means glucose FADH2 becomes or gets converted into FAD. That means it gets converted or it gets oxidized into FAD. And what happens is, because this oxygen will get reduced because it has accepted the electrons from FADH2, it will get reduced and it will be forming H2O. That means this O2 has reduced and this glucose oxidase has oxidized. Okay, so I believe you would have understood what was the reaction that was taking place. Now, glucose oxidase is a basically dimeric protein. Basically, it has two subunits, dimeric protein. Next, two identical subunits are present in case of glucose oxidase. Next, one domain is basically binding to beta D glucose and the second domain is binding to FAD. Since there are two domains, one domain will be binding to beta D glucose and the other domain will be binding to FAD. Beta D glucose means this glucose. So one domain of glucose oxidase will be binding to this glucose and the other domain, this will be binding to FAD cofactor. Okay, now if you understood this, let us move forward. So this is again a diagrammatic representation of what is happening. So glucose is getting converted into gluconic acid. Next, further oxidation process, and oxygen O2 is getting converted into H2O. You can see this. So this is basically what is happening in case of a biosensor, glucose biosensor. Now there are three different transducers that can be used for measuring glucose. Three types of transducers can be used. The basic mechanism of the bioreceptor, the biosensor will remain the same. Depending upon the transducers, you can categorize the bioreceptor or the glucose biosensor into three types. First one is an oxygen sensor that measures oxygen concentration. So one of the transducers can understand or can measure the changes in the oxygen concentration. What oxygen concentration? Now since you saw that oxygen will be interacting with glucose oxidase that was reduced FADH2 form, so what will happen? Oxygen's concentration will be reduced and this particular transducer will be measuring how much concentration of oxygen got reduced and therefore it can predict that what is the glucose concentration. This is the concept. Next is a pH sensor that measures the acid, because I just showed you that glucose is getting oxidized into gluconic acid, right? That means COOH group would be present. Initially I said that a gluconolactone will be produced. Gluconolactone means double bond O. Then it will be converting into COOH, gluconic acid. Previously glucose was OH only. Glucose has multiple OH groups, then converted into gluconolactone, then converted into gluconic acid. Let me just write it down for you. Gluconic acid. Okay, so this was gluconolactone, and this was glucose. Okay, so this is clear. I believe this you would have understood. So pH sensor that measures the acid, gluconic acid, a reaction product. So this is gluconic acid was the reaction product that we just saw. Next, a peroxidase sensor that can measure H2O2 concentration. Now, what happens is often oxygen is basically reduced, and during the process of reduction, it can also form the H2O2, hydrogen peroxide. Now, that can also be detected with the help of a transducer. So, a transducer, a peroxidase sensor that can measure the H2O2 concentration, that can also be used. So, this was basically the different types of biosensors. How does biosensor function? What are the different types of transducers? How the transducers work? Everything. Okay, so I believe this particular video was useful for you. If you liked the video, please hit the like button. Do not forget to subscribe the channel. Thank you so much.